Good evening, everybody. We are back with the current affairs series lecture series that we have been doing. आज थोड़ा कलर का पैटर्न चेंज हुआ है बिकॉज आई वांटेड ये मोनोटोनिसिटी में थोड़ा चेंज आए. तो again we have taken some very important topics for this week of August, like the third week of August, and we'll be concluding the next week in the next session. Now the topic that we have taken today are having very high relevance from prelims as well as the mains point of view, and that is why we'll be going to focus on them in a bit of detail, because questions बहुत prelims में बहुत easily form हो सकते हैं, and we'll be taking up the kind of questions that can be formed. Okay, like how many type of questions can be formed from these topics and all. ठीक है, तो because our aim is to like prepare a subject or prepare some content in such a manner that you are able to do more and more number of questions in the exam. That is the like main aim, apart from imparting the knowledge. So the first one that we have taken is DNA vaccine by India. Why this is in news? Because India has approved the world's first DNA vaccine. Okay, DNA based vaccine for COVID-19. It is manufactured by Zydus. Zydus is a company that is based in India, and the name of the vaccine is CoV-D vaccine. It is based on DNA molecule. Now there has been a lot of speculation about what is the vaccination, how does vaccination function, and what is immunization, how does our body respond to a pathogen that we are infected to. So we have taken all these issues in this lecture, and we will be dealing with all the whole process of immunization, how our body gains immunity, how antibodies function in a very bit of detail, not in very like medical terminology, in such a way that we'll be able to grasp the content key, what it is and how immunization works. Okay. The company has claimed that it will be a 3-dose vaccine built on plasmid DNA. Plasmid is not very important. What we are focused about is, from our perspective, what is important is DNA. Okay. We are using DNA. The efficacy rate of the vaccine is 66.6% against the symptoms of COVID. And one more important thing is that this vaccine is also effective against like pediatric COVID if that happens, because from the age of 12 and above, the children will be having 66.6% efficacy with the similar kind of side effects that adults are having. Okay? So that are like manageable side effects. That is why this vaccine is a very like milestone in this whole vaccine development process. Usually vaccine development takes up to 10 years for like one vaccine to get developed. ठीक है, उसके पीछे दो वजह हैं. The first one is कि vaccines that we have been using up till now, okay, they either used a cultured pathogen which was either weakened or dead, or it was a toxic it was a toxoid based vaccine, like tetanus vaccines, a toxoid based vaccine. अब ये toxoid based vaccine और pathogen based vaccine, ये difference क्या है? We will be looking at all these issues from a very explanatory point of view in a while. ठीक है, तो I hope you understood why we are studying this. Vaccine story so far has been that Sputnik has been developed by Russia. Serum Institute of India has developed COVID Shield, and Covaxin has been developed by Bharat Biotech. Covaxin has been found the Covaxin vaccine का जो immunization benefits हैं, they have been found in around 97 variants of COVID. ठीक है, तो Covaxin of course is a very potent vaccine. Of course all these vaccines are very potent, very helpful, and very like milestones in our whole medical history. ठीक है, तो Sputnik has been made by Russia. ठीक है, COVID Shield has been made by Serum Institute of India. It is a private serum or like pharmaceutical company that is based in Pune, and the other one is Bharat Biotech. Bharat Biotech is a public sector unit. Okay. Now, how vaccines work? We are going to take like the whole lecture about this vaccination process is divided into like 3-4 parts. The first one is how vaccines work, like what kind of vaccines are there, what is the difference between the older methods that have been developed for the vaccination process and the newer ones are based on DNA and mRNA, and the older ones are based on weakened pathogen or dead pathogen or attenuated pathogen, ठीक है, or based on some toxoid like tetanus, ठीक है. So first of all, we'll be taking how vaccines work. First, the most important point is vaccines are not vaccines are not going to cure you if you have encountered any disease. Vaccine is not a curative method, it is a preventive method. Preventive matlab it will prevent you from getting infected. Once you have got infected, it will not be administered to you to get you cured, ठीक है. उसमें दवाइयां एडमिनिस्टर की जाती हैं, ठीक है. बट वैक्सीन लाइक रेबीज का वैक्सीन फॉर बाइट्स अगेंस्ट रेबीज वायरस, ठीक है, और मीजल्स का वैक्सीन, ठीक है, डिप्थीरिया का वैक्सीन, टेटनस का वैक्सीन, ये सारी वैक्सीन्स ऑलरेडी हमारे पास प्रेजेंट है. तो ये कोविड की वैक्सीन, हाउ इज दैट वैक्सीन डिफरेंट? ओके, द कोविड वैक्सीन दैट वी आर यूजिंग टुडे आर नॉट द सेम वैक्सीन दैट वी हैव बीन यूजिंग अर्लियर, ठीक है. फर्स्ट ऑफ ऑल, we should not get hurried into the topic, but we shall be taking it in a bit slow manner so that it is easily understandable. Now vaccines help to body to create or trigger immune response. Immune response means vaccines are basically not acting against any pathogen or not acting against any external body. They are only helping our body create an immune response. Immune response means our body is going to use its own defense mechanism to counter any pathogen. Now what is pathogen? Pathogen can be virus, bacteria, or any kind of external body that causes some disease to us, ठीक है. तो what are like some examples of pathogen? Coronavirus, ठीक है, it's a virus pathogen, ठीक है, और Mycobacterium tuberculosis, the pathogen for tuberculosis. Similarly, अब ये सब जो पैथोजन है, when they attack us, है ना, तो what our body does is, our body has an inbuilt immune response system that is composed of two kind of cells. The first one is B lymphocytes and the second one is T lymphocytes. That we'll be dealing in the next slides. The immune response creates memory cells, ठीक है, memory T cells. Basically, what happens is, when you are affected by a pathogen, ठीक है, our body creates an immune response and remembers it. And whenever you are, that is called a primary immune response. Means for when a immune response has been created for the first time. But when you get infected for the second time or for the third time, the body remembers that this pathogen has affected me some time back, and now I have developed a certain kind of defense mechanism against this virus or pathogen, and now I need to act against it in such a manner that I have developed earlier. That is called secondary immune response, ठीक है. Secondary immune response मतलब अगर आप once you get infected with some pathogen, ठीक है, तो आपकी बॉडी का जो immune response होगा वो primary immune response होगा. When you get infected with the same pathogen again, then your body will be generating a secondary immune response or tertiary immune response based on which time you are getting infected, ठीक है. I hope you understood कि vaccines are not medicines. They are not curative medicines. They are preventive medicines. Okay. तो अब vaccines, when vaccines work, vaccines accelerate our own immune system, then how do they work? They work on our own immunity, ठीक है. Now we'll be going to take two kinds of immunity. Immunity is defined in two versions. The first one is innate immunity. The second one is acquired immunity. Innate immunity का meaning वो immunity है. Okay, I shall be moving this side so that it's clear to you. Now innate immunity is not a pathogen specific immunity. It is not a pathogen specific immunity. It is available to us when we are born. It is accomplished by providing different type of barriers to the entry of a pathogen. Like innate immunity का एक बहुत basic example है हमारी skin. Our skin helps us from preventing many pathogens enter our body. That is like basic defense mechanism of our body against some external pathogen or any external body. So that will comprise of an innate immunity. But it provides basic defense against the pathogens, ठीक है. one such innate immunity is ciliary. Ciliary is very small organelle in our body, tissue kind of, in our lungs, that helps in removing the dirt and dust from our alveoli. Okay? So that is a part of innate immunity. Now if we talk about acquired immunity, okay, this acquired immunity means the immunity that we acquire in our life once we get affected or infected with some virus or some pathogen. तो acquired immunity कौन सी immunity होगी? That we do not have at birth, but when we get infected, then we gradually acquire that kind of immunity. That is called acquired immunity. Understood? It's characterized by memory cells. यानी a very important part about our acquired immunity is that once we get infected, ठीक है, like once some virus, okay, once some virus infects us, ठीक है, ठीक है, once some virus infects us, like this is our human body, ठीक है, let's assume this is our human body, like in remotest of any kind of representation, this will not be the human body, although this is a very similar representation of a virus, but this is not. Still, let's consider this humans, ठीक है. So what happens is when this virus attacks us, okay, so what happens, our body initiates a response against this virus, okay, and then destroys or kills it, and then also makes some copies of its own defense cells, ठीक है, in its own defense cells. These are called T cells. ठीक है, T cells and B cells are two kind of immunity cells. ठीक है, अब B cell क्या होता है? B cell is basically a kind of a tissue. It's a lymphocyte. Lymphocyte we have studied from very childhood that WBCs, white blood cells, are our defense mechanism. White blood cells are basically composed of lymphocytes. Lymphocytes are nothing but B and T kind of. ठीक है, अब lymphocytes B and T हम कैसे classify करते हैं? B lymphocytes are those lymphocytes, ठीक है, those lymphocytes which are formed in bone marrow. ठीक है, these are formed in bone marrow. Bone marrow में और कौन सी चीज का formation होता है? Blood cell, RBCs. ठीक है. T lymphocytes, on the other hand, T lymphocytes are formed in where the thymus, basically they are formed, they generate in bone marrow, but they need thymus to what? Grow and regenerate. That is why they are called T lymphocytes, because they are generated in thymus. Okay. अब thymus is a very small organ situated here. Of course, इसके मेरे rib cage के अंदर यहाँ पे at this point this is situated just here, thymus. अब thymus में T lymphocytes और bone marrow में B lymphocytes. ये दो kind of lymphocytes क्या है हमारा? Defense mechanism. These two cells compose our defense mechanism. ठीक है? Okay. Back to the lecture. I hope you understood कि acquired immunity, innate immunity और acquired immunity में क्या difference है. Innate immunity, the immunity we are born with, is a basic defense mechanism. It is not advanced version. But acquired immunity is adaptive in nature. It means our body acquires or adapts itself to some kind of pathogen when it gets infected to it. And how does that like acquisition of immunity is done, or how that adaptation of our body against some pathogen is done? It is done through what? Memory cells of our T lymphocytes and B lymphocyte cells. Okay? I hope you understood this. This was the basic explanation of how our immunity works. Okay. Now, elements of acquired immunity, as I said, B lymphocytes and T lymphocytes forms in bone marrow, helps in fighting infection against. Now, very important point is B lymphocytes are formed in bone marrow. Bone marrow में RBCs भी बनते हैं. That means B lymphocytes will be circulating in our blood system. Okay, that is why any kind, और यहाँ पे एक और important point जो हमें note करने के लिए है, that B lymphocytes since they are formed in bone marrow, ठीक है, और bone marrow में ही क्या बनता है? RBCs बनते हैं. Okay, तो RBCs क्या है हमारा? Blood या circulatory system का like foundation RBCs है, ठीक है, हमारा blood पूरा circulate होता है हमारी body में. That means B lymphocytes will also be flowing along with the RBCs into this into this circulatory system. ठीक है, circulatory system means, circulatory system कौन सा है हमारा? Blood flow की जो जितनी भी हमारी body में नसें हैं, heart है, that is all composed of that all comprises of what? Circulatory system. Okay, अब circulatory system में क्या है? B lymphocytes. B lymphocyte कोई rocket साइंस नहीं है. B lymphocyte एक सेल है. जैसे RBC is a red blood cell, वैसे ही B lymphocyte is a kind of cell. It is kind of tissue. ठीक है, अगर B lymphocyte सेल, अगर बहुत सारे सेल हो जाएंगे तो टिश्यू ऑफ कोर्स, बट वी विल कंसीडर सेल. जिससे रेड ब्लड कॉर्पसल्स आर RBCs which carry oxygen sorry throughout our blood. ठीक है, throughout our body basically. अब वैसे ही बी लिम्फोसाइट्स का काम क्या है? अटैक करना. किस चीज को अटैक करना? एनी फॉरेन बॉडी. If B lymphocyte detect any kind of foreign intrusion, like if you get infected with some tuberculosis virus, तो it enters your blood system through your lungs. तो blood में ये detect कर लेगा. B lymphocytes will detect कि there is a foreign body and that is Mycobacterium tuberculosis, and then it will start attacking it. ठीक है, अब ये अटैक करना शुरू कर देगा. तो what it will form? First of all, it will destroy. जितनी ये डिस्ट्रॉय कर पाएगा उतने सेल्स. अगर if you are not infected with a huge amount of virus, ठीक है, and if your body's immunity system is strong, then what will happen? कि आपका body B lymphocytes, आपकी body immunity strong है, तो that means you have enough amount of B lymphocytes and healthy B lymphocytes that are actually capable of attacking the tuberculosis virus, and then what happens is your body forms an innate, like acquired immunity against tuberculosis. Understood? This is the basic phenomenon. Now, how does it happens? Whenever B lymphocytes detects an antigen या pathogen का meaning any virus, bacteria, protozoa, any kind of living organism that carries diseases for us. ठीक है, हमारे body में बहुत सारे bacteria or like millions of bacteria and viruses are there living inside in our body, but they are called good because they help us. ठीक है, like हमारी gut में, intestine में बहुत सारे bacteria हैं that help in assimilation of some kind of one kind of bacteria also helps in formation of vitamin K in our intestines. So similarly, there are a lot of bacteria that are very helpful. But pathogen कौन सा हो जाएगा? वही bacteria which causes disease to us, like Mycobacterium tuberculosis. Any virus that causes disease to us is also a pathogen. Any living organism that causes disease to us when it comes in contact with us, that will be a pathogen. अब ये antibody और antigen क्या है? Okay, now हम अपनी समझ के लिए ये कहते हैं कि that is a pathogen, that bacteria, virus, protozoa, whatever it is a pathogen because it is causing disease to us. But when we deal with this whole immunity system, so what we call that external body is antigen. ठीक है, whenever an antigen is detected, so our B lymphocytes come into action and they start secreting antibodies in the bloodstream. Now antibody और antigen में difference क्या है? Antigen कोई भी external body है, antibody उस external body के response में हमारा immune system का जो special kind of defense mechanism है वो antibody है. It is simple. I hope you understood this. Now ये तो हो गया, ये सारा system होता है हमारे blood mediated. Blood mediated, that is why it is also called humerus mediated or humerus mediated humor medium. Humor medium मतलब liquid या fluid medium या blood medium को हम humor medium भी कहते हैं. तो that is why it is called humor mediated response, immune response system. On the other hand, T lymphocytes do not secrete antibodies. यहीं पे एक prelims का question बहुत basic बन सकता है कि एक स्टेटमेंट होगा कि B lymphocytes are formed in bone marrow. सेकंड स्टेटमेंट होगा T lymphocytes release antibodies to fight any pathogen. अब ये दो स्टेटमेंट होगी, आपको चूज करना होगा which one of the following is correct, like one only, two only, both या none. तो ऑफ कोर्स बोन मैरो में B lymphocytes फॉर्म होती है, स्टेटमेंट वन इज करेक्ट. Do T lymphocytes use antibodies? No. T lymphocytes do not generate antibodies. Hence, second statement is not correct. I hope you understood that. तो T lymphocytes antibodies क्यों नहीं बनाती? क्यों? क्योंकि T lymphocytes are formed in thymus and they are found in our nodal gland, lymphatic glands. ठीक है, lymphatic glands हमारे अंडर आर्म्स में है और हमारे पैरों में है. ठीक है, और हमारी बॉडी में बहुत सारी जगह पे लिम्फेटिक ग्लैंड्स हैं। अब ये लिम्फेटिक ग्लैंड्स आर वेरी स्मॉलर ग्लैंड्स जहां पे हमारी टी लिम्फोसाइट्स, मैच्योर टी लिम्फोसाइट्स एग्जिस्ट करती हैं। तो व्हाट हैपेंस इज टी लिम्फोसाइट्स डू नॉट manufacture antibodies by themselves. They rather help B lymphocytes to manufacture any kind of antibodies, or they themselves can attack any pathogen या antigen, and that is how T lymphocytes work. T lymphocytes can themselves either attack any pathogen, or they can help B lymphocyte in formation of antibodies to counter a antigen. But T lymphocytes do not on their own emit antibodies. I hope you understood the basics of T lymphocytes and B lymphocytes. Many people will think that this is too detailed. No, it is all given in NCERT of 12th class. ठीक है, तो yes, and that forms a part of our syllabus. Fine, because वहाँ से questions पहले भी आए हुए हैं biology की जो किताब है. Now this is thymus. It is like somewhere around here. मैंने शायद गलत पॉइंट कर दिया था ऊपर बहुत ज्यादा, बट यस दिस इज थाइमस. यहाँ पे टी लिम्फोसाइट्स का फॉर्मेशन होता है. एंड वी ऑल नो बोन मैरो कहाँ है हमारी, थाई जो फीमर बोन है वहां पे बोन मैरो है, वहां पे आरबीसीज और बी लिम्फोसाइट्स का फॉर्मेशन होता है. तो आई होप यू अंडरस्टूड टू वेरी इंपॉर्टेंट थिंग्स. बोन मैरो का फॉर्मेशन होता है, सॉरी, बी लिम्फोसाइट्स का फॉर्मेशन होता है बोन मैरो में. टी लिम्फोसाइट्स का फॉर्मेशन होता है कहाँ पे, थाइमस में, थाइमस ग्लैंड. एंड द सेकेंड इंपॉर्टेंट पॉइंट इज B lymphocytes use antibodies to counter a pathogen. T lymphocytes do not use antibodies to counter a pathogen. That is the basic of our immune response system. Okay. तो vaccination, how does vaccination work? Vaccination का process क्या है? Now, if you look at this is a measles virus. ठीक है? If our body already has an existing antibody, ठीक है? This is an antigen. ठीक है? अब हमारे ब्लड में अगर ऑलरेडी हमारी बॉडी में एंटीबॉडी है तो इट विल अटैक इट. सिमिलरली चिकन पॉक्स के लिए हमारी बॉडी में एंटीबॉडी है. अब ये एंटीबॉडी हमने वैक्सीनेशन से डेवलप की हो सकती है, ठीक है, या हमारा प्राइमरी रिस्पॉन्स से भी डेवलप हुआ हो सकता है. प्राइमरी रिस्पॉन्स मतलब वी गॉट इन्फेक्टेड समहाउ टू दीज वायरसेस एंड देन आवर बॉडी डेवलप्ड अ रिस्पॉन्स, इम्यून रिस्पॉन्स सिस्टम टू दीज वायरसेस. बट फॉर दैट, for us to develop a primary response system, दो चीजें इंपॉर्टेंट होनी चाहिए. The first one is कि existing attack or existing antigen that we have contracted, that should not be very strong, और उसका अमाउंट इतना ज्यादा नहीं होना चाहिए कि that overcomes our immunity system. ठीक है, अदरवाइज हमें किसी दवाई की जरूरत न पड़ती. We would have been not needing any medicines if our immune system were actually so strong. तो what happened is आपके पास जिन जिन वायरसेस के या जिन जिन पैथोजेंस के एग्जिस्टिंग एंटीबॉडीज हैं, these antibodies will counter these pathogens. But what if you are get infected with a virus or a pathogen or an antigen, no, we should call rather a pathogen. Will be like I'll be telling why we should call pathogen at this point in time. If what happens is if you get infected by this virus or this pathogen and you don't have any kind of antibody for this, so what will happen? तो you might get infected, या अदरवाइज आपकी बॉडी उस अगर आपकी बॉडी आइडेंटिफाई करके और एक नया इम्यून रिस्पॉन्स डेवलप कर लेती है और मेमोरी सेल्स डेवलप कर लेती है उस इम्यून रिस्पॉन्स की, तो whenever you get infected with the next time, तो what will happen? You will be able to counter that. Now what actually vaccine does? ठीक है, a vaccine is a tiny weakened non-dangerous fragment. What happens is, see, this is a pathogen. Yeah, let's take this pathogen. ठीक है, let's take this pathogen. Okay, so what happens is that this pathogen is having these spike proteins. These are called spike proteins. ठीक है, spike proteins. Okay, these spike proteins, और ये इसका for example it is a virus, है ना, and this is its nucleus and genetic material and all that is we are not concerned about. What happens is कि this virus basically or this antigen, this antibody, these are called binding sites. These proteins, spike proteins, are called binding sites. Why? Because when this pathogen infects us, when this pathogen attacks humans, what it does is it binds to our healthy cells. कैसे? ये जो ये इनकी बाइंडिंग लोकेशंस होती हैं, ठीक है, ठीक है. तो फॉर एग्जांपल दिस इज अ हेल्दी ह्यूमन सेल, ठीक है, ठीक है. तो व्हाट हैपेंस इज दिस अटैचेस इटसेल्फ टू दिस पॉइंट, है ना, और या तो अपना जेनेटिक कंटेंट इस सेल में इंजेक्ट कर देगा या इट विल डिस्ट्रॉय दिस सेल. These are the two kind of attacks that a pathogen has on humans. अब हम क्या करें, हमारे पास दो टेक्निक्स हैं. Either we take this spike protein, okay, okay, we'll be discussing that in detail. Okay, हाँ. तो what we do in vaccination is, we take the spike protein, ठीक है. Why don't we have 2-3 methods basically that can be used to initiate a vaccine, like initiate a immune response in our body. देखो वैक्सीनेशन का पूरा प्रोसेस सिंपल है, बहुत सिंपल है. वैक्सीनेशन की पूरी फिलॉसफी इतनी है, कि what we should do is, हमें vaccination is not a medicine to cure anything, it is a preventive medicine, ठीक है. And how vaccination works is कि we have our healthy cells, ठीक है. What we do is हम अपने बॉडी में प्राइमरी इम्यून रिस्पॉन्स डेवलप करने की कोशिश करते हैं. what we try to do is we try to develop a primary immune response, ठीक है. बट ये हमारी बॉडी है एंड इट हैज नॉट बीन इन्फेक्टेड विद अ वायरस. तो जब तक आप किसी वायरस से इन्फेक्ट नहीं होंगे तब तक आपके you would not be having any kind of immune response against that pathogen or virus or bacteria, whatever it is. तो what we do is, तो इसके तो अगर अपने आप को इन्फेक्ट कराने का एक तरीका है, ठीक है, कि either you get infected with corona virus और आप 10 दिन 15 दिन हॉस्पिटल में रहे and then your body might start developing antibodies against the coronavirus and you might get फिर नेक्स्ट टाइम आपको इन्फेक्शन नहीं होगा. ये तो एक बहुत सिंपल तरीका है जो बहुत रिस्की है. इसकी बजाय अगर हम क्या करें, कोरोना वायरस की जो स्पाइक प्रोटीन्स हैं, ठीक है, हम क्या करें, या तो कोरोना वायरस को किल कर दें, ठीक, एंड देन इंजेक्ट दैट किल्ड कोरोना वायरस इनटू आवर ओन सेल्स. व्हाट विल हैपन, आवर बॉडी विल डिटेक्ट अ फॉरेन बॉडी, ओके, एंड आवर बॉडी विल डिटेक्ट अ फॉरेन बॉडी, बट नाउ दिस द वायरस हैज बीन किल्ड. तो ये हमें इन्फेक्ट तो कर नहीं पाएगा. दिस वायरस विल नॉट बी हैविंग द पोटेंसी to cause illness, ठीक है. But what will happen is our body will detect a certain kind of virus, coronavirus, and then it will start developing its own antibodies. Okay, antibodies. अब ये antibodies बहुत सारी डेवलप हो गई हमारी बॉडी में, and whenever we are actually infected with a live coronavirus, ठीक है, तो this one was dead और वीकेंड, ठीक है, डेड और वीकेंड, बट दिस वन इज लाइव एंड वेरी मच एक्टिव. तो व्हेन वी गॉट इन्फेक्टेड, वी पर्पसफुली इन्फेक्टेड अवरसेल्व्स विद अ डेड वायरस. तो आवर बॉडी डेवलप्ड एंटीबॉडीज अगेंस्ट द कोरोना वायरस. ओके, अब हमारे बॉडी में पहले से ही बहुत एंटीबॉडीज हैं, which antibodies are the army to fight any kind of infection. Now a live coronavirus tries to infect us. What our antibodies will do? Okay, come, we already have your defense mechanism. We already have the missiles and tanks that we need to destroy you. Okay, and these antibodies will now destroy it. This is the basic phenomena of vaccination. ठीक है, this was the like whole process of vaccination. कि एक healthy human है, ठीक है, हेल्दी ह्यूमन को हम इन्फेक्ट करते हैं किसी डेड या एटीन्यूएटेड वायरस से, ठीक है, एंड देन हमारी बॉडी में एंटीबॉडीज डेवलप हो जाती हैं, एंड व्हेन द एक्चुअल वायरस अटैक्स अस, आवर एंटीबॉडीज किल दैट वायरस. दैट इज हाउ वी प्रिवेंट आवरसेल्व्स फ्रॉम गेटिंग इन्फेक्टेड. दैट इज द होल फिलॉसफी ऑफ वैक्सीनेशन. अंडरस्टूड? नाउ टाइप्स ऑफ वैक्सीन्स, ठीक है, बिकॉज दिस टॉपिक हैज बीन इन न्यूज फॉर 2 years नाउ. And in last year itself, there were around 50 to 60 articles in the Hindu itself in the science and technology corner on the working of vaccines and how vaccines are developed, how RNA vaccine is different, how DNA vaccine is different. That is why I found it very important that I should address the issue. I should address the elephant in the room, and that is why we have brought this whole detailed lecture on vaccination. So I hope you understood our immunity, how vaccination works, what's the philosophy of vaccination, and now we are going to understand what kind of vaccination that we can use. Okay? Like how has science helped us in understanding the vaccination process. So in the previous slide only, we saw how a dead virus, when we infect ourselves with the dead virus purposefully, that is called vaccine. हम vaccine inject करते हैं, तो we are basically injecting the dead viruses, not the coronavirus vaccine. Of course, I'll be telling why coronavirus vaccine is different from others. ठीक है? What in, like, for example, if we deal with polio virus, so we are actually injecting a dead polio or weakened polio virus inside our body. What happens is, when we get infected, that polio virus enters, it is dead, it cannot cause the illness, okay? But it has a certain kind of characteristic. Yeah, it has the chemical composition of being a biological composition of being a virus, polio virus. Our body starts developing antibodies against that external polio virus that we infect ourselves with, which is actually dead or attenuated. उसके बाद our body has enough amount of what antibodies inside our body, and when we actually get infected with that virus, so our antibodies will kill it. This is the philosophy of vaccination. Now type of vaccine development में एक ही concept है. Again, we have to develop a lot of antibodies inside our body. That is the basic phenomena. चाहे वो mRNA हो, चाहे DNA vaccine हो, चाहे toxoid vaccine हो, चाहे conventional vaccines हो, सबका basic method यही है. So we have basically three kind of vaccination methodologies that we can use. The first one is कि using a whole virus. ठीक है? If we use a whole virus, then what will happen is कि if we are using this whole virus, so we are actually either going to kill it or we are actually either going to attenuate it. That means we are like polio virus. Either we are going to kill that polio virus or we are going to weaken it so much that it is not going to cause us illness. ठीक है? ये तो पहला मेथड हुआ. Second method is this one, where the parts of the vaccine, parts of the virus are taken. Parts of the virus मतलब ये binding sites. The thing that we have talked in our previous slides about the binding sites. These are called spike proteins, as we already have told, right? These are called spike protein. It is a very important, ठीक है, spike protein. तो अब इसमें होता क्या है basically कि we are not going to use the whole virus. Instead, we are going to pull out these spike proteins, okay? And these spike proteins are not going to cause illness. These are only binding sites. ये हमारी सेल से अटैच होने वाली जगहें हैं, बस और कुछ नहीं है इनका काम इस वायरस के सेल में. दिस इज अ वायरस सेल. ठीक है? नाउ व्हाट हैपेंस इज कि वी पुल आउट दीज प्रोटीन स्पाइक्स एंड देन वी इंजेक्ट आवरसेल्व्स विद दीज प्रोटीन स्पाइक्स. अब हमारी बॉडी में क्या होगा, व्हाट विल हैपन इज कि आवर like antibodies, our B cells and T cells, they will detect this spike protein and they know when detected this spike protein what to do. What they will do is they will start building antibodies against this virus, not against the spike protein. Why? Because the spike protein is the part which binds to our cells. तो हमारी बॉडी में स्पाइक प्रोटीन डिटेक्ट करने के बाद उस स्पाइक प्रोटीन के कोरस्पॉन्डिंग एक स्पेसिफिक वायरस के लिए एंटीबॉडी डेवलप करने का मैकेनिज्म ऑलरेडी एग्जिस्ट करता है. दैट इज कॉल्ड एक्वायर्ड इम्यूनिटी. अंडरस्टूड? नाउ दैट इज स्पाइक प्रोटीन. वेट, फाइन. तो या तो हमने पूरा वायरस यूज कर लिया, डेड या लाइव, और डेड या एटीन्यूएटेड, अलाइव, फाइन. या तो हमने स्पाइक प्रोटीन यूज कर लिया, फाइन. या तो थर्ड मेथड, हम बहुत ही नॉवेल मेथड, द वन वी आर यूजिंग टुडे, दिस मेथड एंड दीज बोथ आर बीइंग यूज्ड फॉर द मैन्युफैक्चर ऑफ कोविड वैक्सीन्स. ठीक है? यहां पे हमने क्या किया कि वी आर नाइदर गोइंग टू यूज द स्पाइक प्रोटीन, वी आर नॉट गोइंग टू यूज द होल virus, but what we are going to use is the genetic material. Now, why genetic material? How is genetic material going to help? The first misconception that we have in our lives is the people who are not from biology or life sciences background, they consider genetic material as only the element of heredity. No, it has other functions as well. Like if we take a detour, basic detour, ठीक है, तो एक one basic explanation is कि almost all the functions in our body are done by proteins. Almost all the functions, even our enzymes are kind of proteins. तो अगर हमारी बॉडी के सारे फंक्शंस ही प्रोटीन करता है तो प्रोटीन को कौन बनाता है? दैट इज द सेंट्रल डायलेमा. ठीक है, तो एक सेंट्रल डायलेमा ऑफ जो लाइफ है या सेंट्रल डॉगमा ऑफ लाइफ, इट इज कॉल्ड सेंट्रल डॉगमा ऑफ लाइफ. व्हाट इज सेंट्रल डॉगमा ऑफ लाइफ? डॉगमा का मतलब बेसिक लॉ, मतलब इट्स अ फंडामेंटल लॉ दैट इज नॉट गोइंग टू चेंज. The central dogma is DNA. ठीक है, now we are basically going to understand why we are using genetic material for the manufacture of COVID vaccines. Understood? When DNA manufactures itself, it is called replication. And mind it, this has been a question in UPSC कि what is replication is DNA to RNA conversion, DNA to DNA conversion, DNA to RNA to protein conversion. This has been a question in UPSC. That is why this is very important. Now, when DNA replicates itself, that is called replication. ठीक है? Now, if DNA से mRNA, ठीक है, DNA is deoxyribonucleic acid. mRNA is messenger ribonucleic acid. So this is called transcription. Okay? This is called transcription. And from RNA, when protein is synthesized, this is called translation. Even this page can be a prelims question. Why? Because there has been a lot of buzz about what the protein synthesis and mRNA, DNA, इन चीजों के बारे में there has been a lot of news. T cells के बारे में there has been a lot of news, and that is why this lecture is very important for prelims point of view for 2021 at least. Okay. This is the central dogma of life. मतलब DNA से प्रोटीन सिंथेसिस ऐसे होता है, ये है प्रोसेस. दैट मीन्स हमारी बॉडी में प्रोटीन सिंथेसिस का प्रोसेस क्या है? प्रोसेस है डीएनए से एमआरएनए, एमआरएनए से प्रोटीन. मतलब आज वी अंडरस्टूड दैट हाउ प्रोटीन इज फॉर्म्ड इनसाइड आवर बॉडी. अंडरस्टूड? नाउ इफ वी रिमूव दिस, आल्सो सेंट्रल डॉगमा ऑफ लाइफ. ठीक है, तो एज आई सेड इन द वेरी स्टार्टिंग कि प्रोटीन इज वन कंपोनेंट, प्रोटीन इज वन कंपाउंड दैट डज ऑलमोस्ट ऑल द थिंग्स इन आवर बॉडी. ठीक है, तो प्रोटीन, दैट मीन्स अगर हमें हमारी बॉडी में मसल्स बनानी, फॉर एग्जाम्पल वी हैव टू बिल्ड मसल्स इन आवर बॉडी, तो उसके लिए स्पेसिफिक काइंड ऑफ प्रोटीन होगा. इफ वी हैव टू लाइक डेवलप सम काइंड ऑफ एंजाइम्स इन आवर बॉडी, तो उसके लिए सर्टेन काइंड ऑफ प्रोटीन होगा. अंडरस्टूड? अब व्हाट इज लाइक डीएनए? वी वर डीलिंग विद द फैक्ट कि why is protein synthesis important? यही पे एक प्रीलिम्स क्वेश्चन बन जाएगा ऑफ कोर्स कि प्रोटीन कहाँ सिंथेसाइज होता है, वेयर इज द प्रोटीन सिंथेसाइज? तो प्रोटीन्स आर सिंथेसाइज्ड इन राइबोसोम्स. ठीक है, तो राइबोसोम इज द साइट वेयर प्रोटीन इज सिंथेसाइज्ड. हाउ प्रोटीन इज सिंथेसाइज्ड? इट्स अ वेरी सिंपल प्रोसेस. लाइक वी हैव डीएनए, ठीक है, डबल हेलिक्स मॉडल ऑफ जेडी वॉटसन एंड एफएचसी क्रिक. नाउ that can also be a question कि who discovered the double helical model of DNA, and that was J.D. Watson and F.H.C. Crick. They were awarded Nobel Prize for that. Okay, now this is DNA, not very artistically, not very pleasing, but still, okay. This is DNA molecule from the very simplistic assumption we are taking. So what happens is, what is DNA? DNA is nothing but a set of instructions. You can assume that DNA is a set of instructions which carry the instruction for the protein synthesis. ठीक है, like अगर इस part of DNA को हम break कर दें, तो it might contain an instruction for the synthesis of some kind of muscle protein. For example, okay, muscle प्रोटीन. या अगर हम simplistic language में, we are not getting into a lot of details, but yes, this part of DNA can contain instruction for the formation of some enzyme. Okay, enzyme. Similarly, that is why DNA is the most fundamental unit of life. Okay, DNA has the instruction or the codes that help us in what formation, that helps in formation of certain kind of proteins. Now, why we are discussing all this? Because today we are using, like I already dealt with this. Okay, we are dealing with this type of vaccine is developed by genetic material of the pathogen. We are not taking the whole virus, rather we are taking some genetic material. As I said कि genetic material can be used for protein synthesis. और इससे पहले एक स्लाइड में I said that this is a protein, spike protein. Okay, that means this spike is actually a protein. और ये प्रोटीन इस वायरस का हिस्सा है. दैट मीन्स देयर मस्ट बी सम इंफॉर्मेशन कंटेन्ड इन दिस डीएनए. ठीक है, इस वायरस के डीएनए में या आरएनए में वो इंफॉर्मेशन कंटेन्ड होगी दैट विल हेल्प अस टू फॉर्म दिस प्रोटीन, बिकॉज दिस प्रोटीन इज अ पार्ट ऑफ दिस वायरस. ठीक है, दिस प्रोटीन, स्पाइक प्रोटीन इज अ पार्ट ऑफ दिस वायरस. तो हमारी बॉडी में जितना भी प्रोटीन सिंथेसिस हो रहा है दैट इज बीइंग डन थ्रू द इंस्ट्रक्शंस दैट आर encoded in our DNA. Similarly, in this virus also, this spike protein is being synthesized inside this virus by a mechanism through which an instruction is decoded from RNA or DNA and then a spike protein is formed. Understood? That is why we are not even taking spike protein now. We are taking the genetic material, and that is why now it becomes very important for us to develop a DNA based या RNA based vaccine. उससे पहले हमारे लिए सबसे important क्या होगा? The genome sequencing. Genome sequencing. That is why the Chinese scientists, as soon as they decoded the genome of this virus, coronavirus, they published it online for everyone to use. And that is how the vaccine formation have been accelerated. Okay, तो फर्स्ट स्टेप क्या था? जीनोम सीक्वेंसिंग ऑफ द वायरस. इस वायरस के जीनोम का सीक्वेंसिंग करना सो दैट वी कुड अंडरस्टैंड कि व्हाट इंस्ट्रक्शन इज सिचुएटेड वेयर, है ना, क्योंकि डीएनए कैन बी मिलियंस ऑफ किलोमीटर्स लॉन्ग. ठीक है, तो अब उसको हमें समझना होगा कि विच सेक्शन ऑफ डीएनए वी नीड टू टेक आउट, कि मतलब इस प्रोटीन के स्पाइक प्रोटीन के सिंथेसिस का डीएनए में इंस्ट्रक्शन कोड कहां लिखा है, ये विल हैव टू नो. फर्स्ट ऑफ ऑल, व्हेन वी नो दिस कि we only have to take this portion out from the DNA and then we can of course form spike proteins, or we can inject this part of DNA, ठीक है, this part of DNA or RNA whatever it is in our human cells. अब होगा क्या? बहुत ही इंटरेस्टिंग है. और सॉरी, how this RNA or DNA vaccine works, it is also very important, ठीक है, and interesting as well. Fine. Now we are not in coronavirus vaccine, we are not injecting ourselves with live or dead or attenuated virus or spike protein or anything. What we are doing is कि this is our healthy cell. Okay, healthy cell. Fine. तो हमारी हेल्दी सेल में ये ह्यूमन का है हेल्दी सेल. ओके, ह्यूमन सेल, इट इज हेल्दी. दिस पर्सन हैज नॉट बीन इन्फेक्टेड विद एनी काइंड ऑफ कोरोना वायरस अभी तक. व्हाट वी विल डू इज कि वी विल टेक अ जेनेटिक कोड. ठीक है, इट कैन बी डीएनए और आरएनए. ठीक है, डीएनए और आरएनए, एंड वी विल इंजेक्ट दिस जेनेटिक कोड इनटू आवर न्यूक्लियस. ह्यूमन सेल में डीएनए आरएनए कहां होता है? न्यूक्लियस में होता है. माइटोकॉन्ड्रिया में भी होता है, बट दैट इज नॉट इंपॉर्टेंट. न्यूक्लियस, माइटोकॉन्ड्रिया में ओनली मैटरनल डीएनए पाया जाता है. दिस कैन आल्सो बी अ क्वेश्चन. ओनली मैटरनल डीएनए इज फाउंड इन माइटोकॉन्ड्रिया, बट जो न्यूक्लियस है हमारे ह्यूमन सेल्स का, उसमें मैटरनल और पैटर्नल डीएनए का एक कॉम्बिनेशन होता है जो कि एक नया डीएनए बनता है. तो दैट इज नॉट इंपॉर्टेंट. बट व्हाट वी विल डू इज कि our human cell also have a mechanism for protein synthesis, right? So what we will do, we will inject ourselves with this. What we'll do, we will be, when we are going to take coronavirus vaccine, we are basically going mRNA vaccine, for example, we are injecting ourselves with this genetic material. When this genetic material enters into our body, like in our cell, it hijacks our own protein synthesis mechanism. What it does is it binds to our own DNA. ये हमारे एक DNA का, जो हमारा DNA है, ठीक है, it binds to it, ठीक है, ठीक है. For example, this red part, ठीक है, this red part was human DNA. For example, this red part in this whole sequence was human DNA. What we did is we infected this, ठीक है. If we consider this blue, ठीक है, तो ये ब्लू वाले पार्ट को हमने, जो वायरस का डीएनए है, और वायरस का डीएनए नहीं है, वायरस के डीएनए का एक सेगमेंट है, और ये सेगमेंट क्या बताता है, ये सेगमेंट में इंस्ट्रक्शंस हैं स्पाइक प्रोटीन बनाने के, स्पाइक प्रोटीन्स. ओके, तो व्हाट वी डू इज ह्यूमन डीएनए, ओके, ह्यूमन डीएनए में ही हम ये वाला डीएनए इंजेक्ट कर लेते हैं बेसिकली, एंड देन व्हाट हैपेंस इज कि when this DNA is being used for protein synthesis in our own human cells, so this DNA will start making proteins, spike protein of the virus on our own cells. हमारी ही अपनी सेल्स पे ये वायरस का स्पाइक प्रोटीन बनाना शुरू कर देगा, राइट. अब ऐसा नहीं कि हमारी पूरी ह्यूमन बॉडी की सारी सेल्स में कोई ये इन्फेक्ट करेगा, नो, सम सर्टेन सेल्स, ठीक है. तो अब ये स्पाइक प्रोटीन व्हेन आवर बॉडी डिटेक्ट्स, अगेन नाउ although our cell is human cell, but what is now it doing? It is now manufacturing the spike protein of a virus, of coronavirus basically. Now this spike protein belongs to coronavirus. Our immune cells, basically T cells and B cells, will detect it and start building antibody. They will think that okay, this whole cell is a foreign body, it's a basically coronavirus. They will start making antibodies against our own cell, which is actually not a coronavirus cell. It only has a characteristic of coronavirus cell, ठीक है, and that characteristic is this spike protein. इस प्रोटीन को देखते ही T cells and B cells will start making what? Antibodies. And that is how our whole, हमारी बॉडी में भी बहुत सारी एंटीबॉडीज हो जाएंगी without even infecting ourselves with what? Coronavirus. And that is how our immunity system will start building up. I hope you understood. So again back to this, I hope you understood ki how we were using this genetic material of the virus to hijack our own protein synthesis mechanism, so that our own body starts producing such proteins that are actually representing the spike of the virus itself. And then our body will detect a foreign body and will attack and start building antibodies in our body. तो जो अभी Pfizer, BioNTech and Moderna की जो vaccines हैं, they are all mRNA vaccines. Similarly, the vaccine that we were talking about, that Zydus company has developed CoV-D vaccine that is based on the DNA material. तो आरएनए और डीएनए मटेरियल में कोई बेसिक डिफरेंस, मतलब बेसिक डिफरेंस है, बहुत लाइक परमानेंट डिफरेंस नहीं है. दिस इज द मॉडल ऑफ कोरोना वायरस. एज यू कैन सी, दिस इज द आउटर लेयर ऑफ द रेड कलर, दैट इज द आउटर लेयर ऑफ द कोरोना वायरस, एंड दीज ब्लू sites, these are the binding sites. These sites are basically binding sites, and these are the spike proteins. Okay, the one we were talking about. These are spike proteins. This is what we try to manufacture within our own cell by the use of DNA or RNA virus. Understood? DNA versus RNA vaccine. DNA vaccine is more stable because DNA molecule itself is more stable. ठीक है? और RNA vaccine is less stable because of course the RNA molecule itself is less stable. It can be stored at relatively higher temperatures. it needs excessively low temperatures, and that makes the logistics difficult. Transportation, storage, and these all processes become difficult and costly, okay, for RNA vaccine. It needs a carrier virus. Now DNA cannot just be transplanted into someone. DNA needs a carrier virus which is not very which is not going to infect us. So, okay, so we are going to use some kind of virus which is not harmful to us, but we are taking the genetic material out of that virus. We are injecting this DNA into that virus, and that virus will be used as a carrier, and that we are injecting that ourselves with that virus. Need booster shots. Why? Because when our body detects that foreign agent, that is a virus, we are carrying that DNA of coronavirus into another virus. So what our body will do is, our body will gradually develop immunity against the virus itself. Okay, तो इसके लिए मे बी डीएनए वायरस, अगर डीएनए वैक्सीन की इम्यूनिटी मे बी आफ्टर अ पॉइंट ऑफ टाइम खत्म होना शुरू हो जाए, इसीलिए वी नीड बूस्टर शॉट्स, बिकॉज द DNA molecule is being carried inside a virus which is not harmful to us, but still our body will detect it as a foreign body and start developing antibodies against the virus itself. ठीक है. On the other hand, RNA needs a very fatty like RNA is a very unstable molecule. It is single stranded. ठीक है. और ये क्या होता है, it needs a fatty cover to be transferred to the body. That is why, और इसकी स्टेबिलिटी को मेंटेन रखने के लिए बहुत लो टेम्परेचर चाहिए होते हैं. That is one of the drawbacks of RNA vaccine. Understood? So this was all about RNA DNA vaccine. This is not important. I hope you understood this. If you have any problems, any queries, any comments about this, please mention that in the comment section. I'll be more than happy to take, and this is very important from Clint's point of view. I hope you understood this, and I ask you to go through this video once again, so that you can understand it better, and you can get on to those points that we discussed, which were important for prelims. Okay.